Welcome to the Explainer. Today we're looking at what I can only describe as a real-life biological miracle. We are going to explore how a microscopic organism, one that was actually discovered in the heavily radioactive ruins of the worst nuclear disaster in human history, might literally hold the key to revolutionizing interplanetary spaceflight. So, if you're fascinated by astrobiology, extreme survival mechanisms, and exactly how we're going to pull off the future of human space exploration, well, you are absolutely in the right place. Okay, let's dive into this because honestly, it sounds like pure science fiction, right? I mean, how on earth could the deadly, highly toxic aftermath of a nuclear meltdown provide the exact solution we need to keep humans alive in the harsh, unforgiving environment of deep space? Well, the answer lies in the just incredible adaptability of life. Specifically, we're talking about extremophile organisms that don't just happen to survive in lethal conditions, but they actually actively thrive in them. So, here is our roadmap for today. We're going to cover the initial Chernobyl fungi discovery, how these fungi actually go about eating radiation, the results of testing these microbes in space, how we can use them for biomining and radiation shields, and finally, the next steps for deep space exploration. Chapter 1. The Chernobyl Fungi Discovery. Let's move to and see how this builds. Because to really understand the future of spaceflight, we have to step back into the eerie, highly dangerous aftermath of the 1986 reactor meltdown. Our timeline of human understanding here begins in 1991, when scientists first noticed something bizarre in those ruins. By 2007, laboratory breakthroughs had revealed the incredible biological mechanics at play. This ultimately led to a commercial patent being awarded in 2014. And by 2018, these exact same microorganisms were actually being launched into orbit for experiments aboard the International Space Station. So let's go back and set the scene in 1991. We are 5 years out from the disaster. Scientists who were sending remote robots into the heavily contaminated zones of the Chernobyl Nuclear Power Plant made a totally shocking discovery. The cooling waters of the reactors had completely turned black. They found over 200 species of melanin-rich fungi just happily thriving in an environment where the radiation levels were completely lethal to humans. Early investigations by scientists like Nelly Zhdanova and her team revealed that this black mold wasn't just surviving the radiation, it actually seemed to be drawn to it. And that brings us to this absolutely stunning statistic. Zhdanova and her team conducted an experiment where they placed fungal samples near radioactively decaying cadmium-109 and phosphorus-32 isotopes. And what they found was that 66.7% of the fungal samples exhibited a behavior known as radiotropism, meaning they actively and intentionally grew in the direction of the radiation source, totally independent of where their normal food base was located. They were quite literally seeking the radiation out. Chapter 2. How Fungi Eat Radiation. This bizarre behavior posed a massive question to microbiologists. How do these organisms survive an environment that so aggressively destroys the DNA of almost every other living thing on Earth? Well, the answer is radiotrophy, which is also called radiosynthesis. Think about it like this. Just like plants use chlorophyll to turn sunlight into energy via photosynthesis, these organisms use melanin, which, by the way, is the exact same pigment found in human skin, to capture deadly ionizing radiation and convert it into usable chemical energy. This allows the fungi to function as autotrophs, meaning they essentially make their own food out of something that would normally kill them. So let's introduce the specific heroes of our biological story. There are 3 key melanized fungal species, and bear with me on the pronunciations here. We've got Cladosporium spherospermum, Weignella dermatitidis, and Cryptococcus neoformans. These specific species were heavily studied back in 2007 by microbiologists Ekateria Dadashova and Arturo Casadevall at the Albert Einstein College of Medicine. They realized that ionizing radiation actually changes the electronic properties of the melanin inside these fungi, which essentially supercharges their growth. Now, what's really interesting about this slide is that the sheer scale of their resilience is just hard to wrap your head around. In the lab, researchers placed these melanin-containing fungi in an environment where the radiation level was a staggering 500 times higher than the normal background environment on Earth. In a setting that should have been instantly lethal, these organisms didn't just survive. They rapidly increased their biomass and accumulated acetate at highly accelerated rates. Looking at the objective data from these experiments, the comparison is just staggering. When scientists compared the growth rates of the fungus Cryptococcus neoformans in a normal, everyday environment versus a heavily irradiated environment, the irradiated fungus actually grew 3 times faster. The ionizing radiation wasn't harming the organism at all, it was acting as a highly efficient food source.
Chapter 3: Testing Microbes in Space. Having proven this in Earth-based laboratories, the scientific community had a massive realization. This could potentially solve one of the greatest hazards of deep space exploration: deadly cosmic rays, coupled with strict resource scarcity. The astrobiological potential of this discovery is heavily documented in rigorous, peer-reviewed literature, like the World Journal of Advanced Healthcare Research. Researchers postulated that, hey, if these fungi harvest energy from radiation, they might be the perfect organisms to bring into low Earth orbit. We could test their resistance to cosmic radiation and extreme stress factors like microgravity and desiccation.
And this led to a truly pivotal NASA experiment. From October 2018 through March 2019, an autonomous experiment was sent all the way up to the International Space Station. Researchers utilized specialized Space Tango flight hardware to cultivate Cladosporium's herospermum, which is a type of dark pigmented or domatoceous fungus right there in low Earth orbit. They exposed it directly to the unique space radiation environment over the course of several months. The results of that mission, which were analyzed in a major study by Schunk and colleagues, were incredibly promising. First off, they confirmed that the fungal growth curve was significantly steeper in space compared to the ground controls back on Earth. But more importantly, they ran statistical robustness tests on radiation attenuation. By placing a radiation detector beneath the growing fungus and another beneath a negative control with no fungus, they found that the radiation levels beneath the fungal bed were demonstrably lower. The fungus was actively blocking and absorbing the incoming cosmic radiation, proving its viability as a living, bioregenerative shield.
Chapter 4: Biomining and Radiation Shields.
So we know they grow really well in space, and we know they block radiation. This opens the door to two massive practical applications for deep space missions: resource extraction and habitat construction. First, let's look at extraction, specifically the BioAsteroid project, which was led by researchers from Cornell and the University of Edinburgh. Basically, astronauts aboard the ISS literally fed pieces of meteorites to microbes. They used the bacterium Sphingomonas desicabilis and the fungus Penicillium simplicimum to extract highly valuable platinum group elements from stony L chondrite meteorites. These microbes release carboxylic acids that bind to the minerals and dissolve them into a solution. This allows astronauts to biomine resources straight from space rocks. And this brilliantly illustrates one of the most surprising findings from lead author Rosa Santamartino's research. When comparing Earth gravity controls to the microgravity environment of the ISS, they found that microgravity actually altered the microbes' metabolism for the better. The lack of gravity increased their production of carboxylic acids, meaning these microbes are actually more efficient at mining metals up in space than they are down here on Earth. How wild is that? Alongside mining, there is the incredible potential for building radiation shields. According to the ISS attenuation data, you don't actually need a massive wall of pure melanin to protect astronauts. The fungal radiation shield tested was composed of 91.4% non-melanized biomass and just 8.6% melanin. That tiny, tiny percentage of melanin does all the heavy lifting, acting as an incredibly efficient self-healing barrier against ionizing radiation.
Chapter 5: Next Steps for Deep Space.
So the crucial point is this: as we look toward the Artemis program's goals of establishing lunar bases by 2028 and eventually launching crewed missions to Mars, solving the payload problem is absolutely essential. Transporting heavy traditional shielding, like lead or tons of water from Earth to Mars, is incredibly costly and, frankly, logistically impractical. Instead, astronauts could use in situ resource utilization, or ISRU. The idea is they transport a lightweight, minimal payload of dormant fungal spores. Once they land on Mars, they simply add local Martian water and nutrients and allow the fungus to grow into a self-replicating, self-healing, living radiation shield right over their habitats. Now, to maintain an objective look at the science, it is important to note the current limitations and ongoing debates in the field. While the process of using melanin for energy was patented back in 2014 by Dadachova and her team, the exact quantum mechanisms of radiosynthesis are still being mathematically modeled today. Astrobiologists also emphasize that we need more long-term studies on how these fungi interact specifically with galactic cosmic rays, as opposed to the localized gamma rays we usually use in Earth-based laboratories. We also really need to better understand how the fungi handle multi-factor stress, like dealing with radiation, microgravity, and desiccation all at the exact same time. But why does all this matter for us right now? Well, the applications beautifully bridge the gap between Earth and space. For astronauts, these compounds could be used as a biological UV prophylaxis, basically the ultimate sunscreen.
Or they could be cultivated as an inexhaustible radiation-fed food source on multi-year missions. And back home on Earth, these radiotrophic fungi offer an incredible potential solution for the bioremediation of our own nuclear waste sites, safely breaking down radioactive contamination over time.
We started in the dark, highly radioactive waters of Chernobyl, and somehow ended up looking at self-growing bioshields on the surface of Mars. It completely forces us to rethink the very boundaries of biology. I mean, think about it. If life on our own planet can adapt to actively feed on the most destructive energy humanity has ever unleashed, what other microscopic miracles are out there waiting to be discovered in the extreme environments of the cosmos? Thanks for joining me on this explainer, and keep questioning the universe around you.